But are there any questions at all related to the first lecture on Tuesday where we went over water balance and homeostasis and really, hopefully, I was able to convince you that sodium is really important, and where sodium and potassium are found within the cell or outside of the cell will be a very important part of our conversation today. And we are also looking at just the overall idea that we have these compartments, that water is found in those different compartments normally, that we can have excess fluid in those compartments, we can have dehydration, and we also looked at the hormonal signals that are involved with driving you to be thirsty to correct those imbalances. So that's the big focus of Tuesday. The Connect assignments, if you haven't already gotten into Connect, please, if you're having any trouble, let me know, but go ahead and get enrolled in Connect tonight, today. If you once you get in there, you will see that there is an assignment for Chapter 1. It's one question, and it's just basically a reminder what anatomy versus physiology is. And then you'll find a Chapter 24 assignment, which will go through the concepts that we talked about on Tuesday. And now today, you'll see for today a Chapter 12 assignment. There will also be on there a quiz. There will be actually 3 quizzes that you will see before the first exam, but only one quiz is now available to you, and it's on water balance. Those quizzes are not timed. They are open book. You can come and go, go in, leave, go back, and finish and move around within the quiz. And just make sure you complete that quiz before the deadline. And as I recall, the deadline is 2 days before our exam. Okay, so I'll go over those dates with you again. Is anyone having trouble with Connect? Getting on to Connect? Okay, talk to me afterwards. Let's get this figured out, okay? Anybody else? Two people over here. Anybody else having trouble? Okay, those who are on, is it working okay? Yep. You're in, you're seeing it? Okay, very good. Well, let's get started then with Chapter 12 if there are no particular questions at this point. Okay, so Chapter 12, we talked about Chapter 12 back in 105. This was the chapter where we introduced the neuron. This is the chapter where we talked about the nervous system as an overall introduction. And we recognize, this is where we're starting, right? We're starting with the nervous system, and our first exam in here will be largely on the nervous system. Our first lab practical will be largely on the nervous system. So this is going to be with us now for the next couple of weeks. So we know the nervous system then is this complex system. It's the one I spent the most time on in 105. We spent 5, maybe 6 chapters in the nervous system. So it's very, very important, and I think it's fundamental, so we'll start here. And you'll be learning about different parts of the brain and a lot more about the brain and the nervous system than perhaps you already know. So overall, and I got this far, right? Last time I'm just reviewing it because I didn't record this on the presentation that I did yesterday. But we know the nervous system is carrying out 3 basic steps, right? It is the system that is receiving information, and that sensory information can be found either in the internal or the external environment. We can have visceral signals coming in, or we can have external signals coming in. The nervous system is going to integrate all of those signals, process all that, figure out what's going on. Most of those signals will never reach our consciousness, right? Most of that integration will occur in our subconsciousness, in our brainstem, or in our spinal, the basic brain areas or the spinal cord. And then we'll send out signals. Again, the signals that are being sent out are largely going to be autonomic, right? Things that we're not voluntarily in control of. If something is important enough for us to know about it, then the signal will go up to our cortex, to our cerebral cortex. We'll make a voluntary decision about it, and then we'll send those signals out as well to the voluntary things. What were the other words we wanted to include here? So for the first half, things that are receiving. What's the other word for receiving? Yeah, afferent, right? And then for those things when we're sending signals out, those are efferent. Good. I'll get the hang of this. The universal characteristics of the nervous system include the neuron being the major cell that we'll be dealing with. Remember that these cells are excitable. Another way of saying that is that they're irritable, sort of like maybe you are in the morning, right? And And that means that they're going to respond to some stimuli. And then there's going to be some conductivity. A signal will be sent. That signal we'll get to know as the action potential. And we'll be studying how exactly that signal is propagated down the axon. And then that axon is oftentimes very, very long, and that signal will be propagated all the way down the axon until we reach to the telodendria, or the axon terminals. And there at the very end, there'll be a synaptic knob, and from there, neurotransmitters will be released, and that will cause a secretion or some effect to occur. Now, what are those neurotransmitters going to affect? They're going to drop off their neurotransmitters to tell a muscle to contract. It could be voluntary muscle, or it could be involuntary muscle, or it's going to tell a gland to secrete, right? And we call those the effectors. So the efferent signals go to the effectors, which are going to be the glands or the muscles that receive these neurotransmitters. Okay, so here we are back to our favorite cell, the neuron. And I've got about 5 or 6 slides here. Some of this should be very, very familiar to you and a reminder of some things you already know about the neuron. Some of these details will be new and improved. So let's think about the neuron. We have the soma, right? The soma is the cell body. You'll see the neurosoma as a word sometimes. And another name for all of this is the perikaryon. Now, what does perikaryon mean to you if you break that word down? Peri, around, karyo, nucleus. The nucleus. So would you agree that everything outside around the nucleus is the cell body? You can also think of it as being the cytoplasm of the soma, okay, the perikaryon. And when you look at a neuron, you see one single nucleus, centrally located. The cytoplasm is going to be very, very rich in mitochondria. The neuron is extremely metabolic. Lots of energy needs of this cell, so there'll be a lot of mitochondria to provide that. Lots of lysosomes as well. Now remember that the neuron is a cell that cannot replace itself, so it better have its own garbage system. And that's what I think about the lysosomes, right? A cell that is rapidly turning over and is being replaced constantly would not need a well-developed garbage system. But the neuron is going to hang around your entire life, so it has to have a well-developed lysosomal system to break things down as they need to be. The Golgi, remember, is a part of the complex that makes proteins along with the endoplasmic reticulum. The Golgi is involved with that. And there will be lots and lots of rough ER. When I say rough ER to you, what comes to mind? It's rough because there are on its surface ribosomes, and ribosomes are primarily involved with protein synthesis, right? So this is a cell that is making buckets of proteins. And what kind of proteins is it making huge numbers of? Neurotransmitters, absolutely, right? So this story all comes back, and remember that no two cells are exactly the same. So if I'm comparing a neuron with a liver cell, they're all going to have the same organelles, but the neuron just simply has a really high percentage of ER and ribosomes, a really high percentage of lysosomes and mitochondria. Now inside this cell, there's going to be some cytoskeletal proteins. This is an oddly shaped cell, isn't it? This isn't just some round ball. This thing has this long axon and all these dendrites, and all that structure is going to be given by some cytoskeletal proteins, proteins inside that are basically giving it structure. And we'll see those. Those are called the neurofibrils, and they're basically microtubules. Remember, the microtubules are similar to what's being sent out for the mitotic spindles and what's involved with flagella and a bunch of other. Whenever you see something sticking out of a cell, you know there's some sort of cytoskeletal protein involved making that happen. Now inside the cytoplasm, inside the perikaryon, one will see Nissl bodies. Now this is just a guy's name, Nissl, and he described, when looking under the light microscope, he saw these blebs, these dark staining areas. And now we know that those Nissl bodies are clumps of ribosomes and clumps of rough ER, right? Because there's such a high density of these protein manufacturing units. Now, another thing about neurons, there are no centrioles. Do you recall what centrioles did in a cell? Centrioles were part of the mitosis, and it was from the centrioles that the mitotic spindles were sent out during mitosis to pull the chromosomes to the daughter cells. Well, neurons don't have centrioles. Therefore, neurons can't undergo mitosis. Therefore, neurons can't divide and make more of themselves. So even if we wanted to tell a neuron to divide, it doesn't have the mechanism inside to do so. It doesn't have the centrioles. There are also glycogen granules. When you hear the word glycogen, hopefully you're thinking, sounds like glucose, sounds like sugar, and it is sort of a sugar kind of molecule. Again, these are highly metabolic cells. They need their own little storage shed of food, and so those glycogen granules are a food source. There's also lipid droplets, and a pigment called lipofuscin. Now, what it is simply when lysosomes start breaking down worn out organelles, we get more of this lipofuscin. I told you that neurons need to have a good garbage system, right? The neuron itself can't be replaced, but the organelles inside of it can be. So as the organelles are being broken down and being recycled, the lysosomes are breaking it down, and you start getting sometimes a sort of a dark pigment within the neuron. Okay, so you'll see more of this lipofuscin as the neuron ages. And again, you'll see more of it in an older neuron. Dendrites. What do we know about dendrites? They're the receiving or the sending part. They're the receiving, typically, the receiving processes. And some neurons will have 10,000 dendrites. Okay, so what we have in our cartoons are usually very simplified diagrams to help us handle an understanding of what's going on here. But some dendrites have some neurons have a vast number of these dendrites. And we know that dend means what? Tree, right? So in 106, I don't have a vocab section of the course, but I do expect you to remember the vocab from 105, okay? So we've been there, we've done that. And so if you took 105 with me, you might want to just, every once in a while, if you made flashcards for vocab, you might want to flash through them every once in a while. I'll continuously remind you of those things as we go through. So again, the dendrites are usually receiving the signals coming in from another neuron. Remember, the information traveling through a neuron is unidirectional. So typically comes in at the dendrites, it can come in directly at the soma, and then continues out and down the axon. So speaking of the axon, the other name for that is the nerve fiber. Okay, you'll hear the term nerve fiber as the same as an axon. So what is a nerve? A nerve is a bundle of axons, or a nerve is a bundle of nerve fibers. Okay, so you'll hear it used both ways. The axon is attached to the soma through a larger conical or triangular shaped area referred to as the axon hillock. Strange little name, but it's the connection point. And the axon, there'll only be one axon coming off from the cell body, but there might be some branches, right? So there can be some collaterals, right? Lateral off to the side, some little branches going off to the side. And this red arrow reminds us that the information traveling is unidirectional, moves down just one direction. Now there can be a lot of branching at the end, and those branches are going to allow one neuron to communicate with many, many cells, right? So it's not just one axon going to one muscle cell, it's one axon that spreads out and has many, many little fingertip ends. Those are called the what? Axon terminals, or the telodendria. It's coming on the next slide. But that allows for one axon to communicate with many, many cells, essentially simultaneously. Okay, a couple more words here. Some, like muscles, have specialized names. We don't say that we say that a muscle cell has a sarcolemma, the outer layer of a muscle cell, and we say that the inside of a muscle cell is sarcoplasm rather than cytoplasm. The same holds true for the neuron. Inside the neuron, you might hear it called axoplasm for the cytoplasm within the axon. Right? This is a very, very long process. So the cytoplasm within it collectively is called the axoplasm. The layer around the axon is referred to as the axolemma. Again, one axon per neuron, and you will see along the axon some Schwann cells. Now, is that always true that you'll see Schwann cells? No. If you're in what nervous system, you'll see Schwann cells? Peripheral. If you're in the peripheral nervous system, you'll see Schwann cells. What I don't have listed here is that if you were in the central nervous system, you would instead see what? Oligodendrocytes. Good. Okay, oligodendrocytes. Now, at the very end, again, the axon will split into a bunch of little axon terminals. Those are sometimes called arborizations. Why arborizations? What do we do on Arbor Day? Plant trees, right? So the arborizations are the same as the axon terminals, and that's also the same as that other term I mentioned briefly, telodendria, right? So dendria, tree, telo at the end, right? Little trees at the end. They're all the same. Unfortunately, they have been named by multiple people different ways, and the literature has used and maintained all three of these terms. What do we find at the end of those axon terminals? Again, we find synaptic knobs filled with synaptic vesicles. When I say the word vesicle, what comes to mind? A little bubble, right? A little bubble. And what's in that bubble? Some neurotransmitters, right? Now, where were those neurotransmitters made? Yeah, by the ribosomes up here, right? Up in the soma. That's where the ribosomes are, right? That's where the rough ER is. So those neurotransmitters are made up here, and then they are going to squiggle their way down the axolemma, contained within vesicles, and be stored down here at the synaptic knobs. And then they will be released as instructed. Okay? So little synaptic vesicles filled with neurotransmitters.
So as soon as they're made, they're immediately transported and stored there?
They are on a journey. Okay. And we'll talk about that journey and how long that journey takes. If we're talking about a neuron that travels from your spinal cord all the way to your big toe, it can be days. This is a highway of it's a really cool highway that we'll talk about. And the rate of how quickly these things can move is rather limited. But you have some neurons that are a meter long, and it takes quite a while. So that means you have a constant making of neurotransmitters up in the soma, and they're being packaged and sent down in little vesicles, little bubbles, down the axon. And we'll talk more about that in a few minutes. Okay, in fact, not in a few minutes, but right now. Okay? So the proteins are made up in the soma, right? They're made up in the cell body, and they're going to be transported down. And along the way, we're going to be having this, what we call axonal, sorry, I'm doing this wrong again. Axonal transport. Okay, now, when I tell you that the electrical signal travels through the neuron unidirectionally, I'm not lying to you, right? So information comes into a neuron through the dendrites, is processed in the soma, and then continues down the axon. And that's true. The electrical signal moves in one direction. But this highway of information going up and down the axon is bidirectional. Now, I'm not talking about electrical messages, but I am talking about chemical messages. Okay? So up and down the axon, there's going to be both what we refer to as an anterograde and a retrograde transport. I'm going to start with anterograde, right? And that's what we think about. It's the movement of things from the soma to the end, to the distal end of the axon. Okay, that's anterograde. And that's the way that most things are moving, right? From the soma down the axon. But things need to be repaired. There's a lot of stuff going on in the neuron that needs to or requires retrograde movement. So there are also going to be some movement of molecules up the axon toward the soma. Now, this highway of movement is mediated along microtubules. I told you a moment ago that microtubules were the cytoskeletal proteins that help to give shape to the long axon, right? And what we're going to see is that there are actually proteins that walk, literally walk along the microtubules. And it creates a highway. And they're carrying molecules, some carrying molecules down the axon, anterograde, and some carrying molecules back up the axon, retrograde. The two proteins that do this movement, kinesin is anterograde. Now, when you hear the word kinetic or kinesiology or kinesin, what comes to mind? Movement, right? So this is a protein that moves, right? Kinesin, I-N on the end, tells you it's a protein. Now, that's the protein that kind of carries things down toward, it's like Santa Claus with a big sack, right? He's carrying the toys down the microtubules toward the soma, or sorry, toward the axon terminals. Now, the protein that carries things the other way is called dynein. Okay, I don't have quite a story for that one, but it's not kinesin, it's dynein, and that's the one that's moving things more retrograde. They are motor proteins, which means that they do what? Move, right? They're motor proteins. They move. They literally slide along the microtubules, right? Slide or walk or move, however you want to think about it. Now, there are different rates. How fast can things move? There's both fast and slow. So, fast axonal transport, 20 to 400 millimeters a day. Okay? So, some of your molecules can move 400 millimeters a day. That's up to, right? That's the speed limit, max. Okay? So this is going to be organelles, right? We've got to move organelles. There are mitochondria, not just up in the cytoplasm. There are mitochondria all the way down at the synaptic knobs. So we've got to move organelles. We've got to move enzymes. We've got to move those synaptic vesicles filled with neurotransmitters. Okay. And so that's fast anterograde. So let's say you have a meter-long neuron. Right? What's a meter? 1,000 what? 1,000 millimeters. So in that case, how long would it take at the fastest rate for a molecule to get from your spinal cord to your big toe? A couple of days, right? And that's the max. It could be a little slower. So we could have a week or a couple of weeks even for molecules to make it all the way down a really long axon. Okay. Now, there's also fast retrograde transport. And this is not by design, but this is the way that rabies gets into you. This is the way that herpes gets into you. This is the way that polio virus gets into the body. So a person is exposed to herpes virus. And upon initial exposure to herpes, there will be a very broad area of infection. So we're talking about simplex 1. You know, a person who just gets herpes for the first time will have sort of a breakout all over their mouth perhaps. And that virus is going to travel up the neurons, retrograde, and go back toward the soma. And that's where it stores. And then when there's a recurring breakout of the herpes, it comes back down, but then is much more localized, right? Much more pinpoint when it comes back down and reinfects over and over. Okay. So, but rabies, when someone gets bit by a rabid animal, the virus makes its way back up to the nervous system and ultimately to the brain, which is why rabies is 99.99% fatal if you do not get treatment, right? If you let that virus get up into the brain, you're dead. So if you get bit by a rabid dog, you better go in and get the antiserum and get fixed, or else there's not much we can do for you once that virus goes back up the nervous system to the brain. Okay? So that's why there's a delay too. Someone gets bit by a rabid dog, it's going to be a couple of weeks before we start seeing symptoms, and by the time you start seeing symptoms, it might be time to call the undertaker, okay, because it might be too late. So, horrible story, but it's true. Now, there's also slow axonal transport, okay? So there are other molecules that go much slower, and this is more axonal flow. It's not like they're being carried really, really fast. It's just sort of like, okay, something kind of going down the river as slowly as it can. And this is always anterograde. There's no retrograde slow. So it's always anterograde. And this is moving other things, repair mechanisms. In a few minutes, I'll mention how neurons can sometimes be repaired. Their rate of repair will be at this rate. Right? They'll be very, very slow. So there's going to be any kind of repair of a neuron, it's going to be very slow at the rate of slow transport. Now neurons, like I'm trying to tell you, are going to stick around in your body your entire life, right? Once they're formed, that's it. They're there for you, so they need to be well protected. So we need to figure out now how we're going to take care of ourselves for decades. Question? It depends on the extent of the damage and how far away the damage is from the muscle or whatever it is that you're connecting to. And the rate of repair is back at this rate, right? So if you're going to have repair, it's going to be about it's going to be back to about a half millimeter per day, maybe 1 millimeter, up to 10, but figure slowly, half to 1 millimeter a day. So the neuron is not being neuron's not being regenerated. Just the axon, if I misspoke a moment ago. So the neuron is still there. It's the axon that's going to repair itself and re-extend itself, but the neuron itself cannot be regenerated or divide. Okay, so one of the things we need to do is protect that neuron as well as help with the electrical signal. So we've got myelin here. It's going to be doing a couple different things. Number one, it's going to insulate the nerve fiber. It's going to insulate the axon. And it's going to do this, the myelin is going to be made by one of two cells. We've already alluded to them. There's the oligodendrocytes, the cells found in the central nervous system, and there are the Schwann cells found in the peripheral nervous system. Now let's make sure we have this word figured out. Oligo. What do we have for oligo? A few, a limited number of trees coming from one cell. So what we'll see in a minute, I'll show you a picture, is that the oligodendrocyte reaches out with a few tree-like branches and helps to myelinate some axons. Okay? And then there are the Schwann cells. Now the Schwann cells have another name that I want to point out to you. I know the other name for those Schwann cells. Neurolemocytes. Okay, neurolemocytes. What's that word mean to us? It's a cell, site, part of the nervous system, right, or around the nerve fiber, the axon. That lemo, that is an outside layer or shell, right? So again, it's the same idea. It's a cell that wraps around the nerve fiber. Now, myelin is a white protein, but it's also considered a lipoprotein. Okay, so that means that it's part protein and part lipid, part fat. Now, it has the IN ending, right? So it's a protein, but it has a lot of fat mixed with it. And this fat is going to protect and insulate not only the axon from damage, but also, we'll see in a few minutes, protect the electrical signal as it travels down the axon. Now, myelin begins in about the 14th week of development. So up through the first trimester, there's really no myelination going on of the nervous system. And it continues all the way through infancy. But it's not completed in some of you, right? It doesn't finish completely until late adolescence. And even there's evidence that part of the frontal lobe is not completely myelinated till about the age of 25. So this is a continual process. And you already know something about myelin. Myelin does what? It helps to speed up electrical signals. And so there are some parts of your brain that are not as well functioning as a young person that they will be later on. The old adage was, old people are wiser. Well, they're also more myelinated, right? So they're wiser because they've got the whole selection of synapses, and they've been there before, and they have a lot to teach you. So listen, okay, that's just my little commercial.
Like if a child was to get hurt or hit their head or whatever, wouldn't we do more damage to them than it would an adult? Like for a child to get hurt, they can always recover.
The brain, a child who gets hurt, they have more plasticity. The brain can recover, and other parts of the brain can make up for that. But what's the rest of your thought?
But compared to an adult is not. So wouldn't it be the opposite, you think, because of the fact that a child isn't as protected as an adult?
Yeah, I see what you're saying, that we're not as myelinated, but also the child is still making new neurons as well and is making new synapses. And so the child has an ability for more plasticity, for more change, okay, because the nervous system isn't as well developed yet. Now, what do we know about the architecture of a Schwann cell? One Schwann cell wraps its entire little body, right, around a portion of an axon. And one Schwann cell wraps around just a portion of one axon, right? That's the architecture of it. So we'll see that it spirals around repeatedly, around and around and around. And there'll be hundreds of layers of this myelin. That outer layer, again, I used this word earlier, the neurolemma, right? The neurolemma is the outside layer, and the Schwann cell is outside of that even, okay? Now, we'll see the architecture of a nerve here in a moment, but there are 3 layers of connective tissue within a nerve. The innermost layer is the endoneurium, and we'll just, in a moment, I'll show you the endoneurium, and it's the layer right around the Schwann cells. So hang in for that. You'll see it better in a moment. So here's the actual axon. We see it traveling down. We can see it traveling down through the center. Here's the Schwann cell. One cell. Well, it looks like about a dozen layers, but there could be hundreds of layers of myelin wrapped around and around. This cell has its own nucleus. This cell can die and be replaced. So Schwann cells are mitotic, and they are constantly replacing themselves as needed. The neurolemma, again, is the outside layer around the whole thing. Remember, the cytoplasm inside would be called the axoplasm, right? The cytoplasm inside the axon itself. There's a space in between the Schwann cells, and that space is called the what? The node of Ranvier, right? Now, if those are the nodes, there'd be a node here and a node here. What would the internodes be? What does inter mean? Between. So, how would you describe the internodal region? The internodal region would be myelinated, right? So the area between the nodes is, in fact, completely surrounded by a Schwann cell. So the nodes of Ranvier are naked little areas. The internodal areas, right, between the nodes are, in fact, covered by a Schwann cell. Now, in the central nervous system, we have, instead, the oligodendrocytes. And again, by the word's derivation, we know that it has a few little branches that reach out, and they anchor not to one, but to multiple axons. And they can't migrate around. Schwann cells have a little bit more movement, if you will. Okay. And that's all I'll say right now, just so you have the oligodendrocytes in the central nervous system. Doing okay so far? So, mostly review, right? Hopefully mostly review. Now, it takes many Schwann cells, end to end, right, to cover the length of an axon. It would also take multiple oligodendrocytes to cover the length of an axon. Remember, we only see myelin where on a neuron? Where do we see myelin on a neuron? Only on the axon. None on the dendrites, none on the soma, and none on the axon terminals. Right? So at the very, very end, where the axon branches into its little tiny branches, there's also no Schwann cell down there. So again, we've already mentioned this. We have the nodes around VA. The internodes would be the areas where there is myelin, where the Schwann cells are, or where the oligodendrocyte is covering. And there is a little area called the initial segment. Can you picture the axon hillock? Right? So you've got the soma, and off the soma you have soma, and then you've got that triangular region, if you will, the axon hillock, and then the axon. So there's going to be a little bit of an area here where there won't be a Schwann cell. Okay? So that's called the initial segment. Makes sense, right? And here I'm using the word glial cell. What's a glial cell? Any of those helper cells. And what did glia mean? Glue. So these are the cells that do what? Kind of glue the nervous system together, give it additional structure and give it additional protection. There's also something else very, very important happening in the axon hillock and in the initial segment. Collectively, the axon hillock and the initial segment are referred to as the trigger zone. The trigger zone. And this is going to be bringing us into this conversation about action potentials. This is where the actual nerve signal, another way of saying nerve signal is action potential. But that's where the action potential is actually going to be initiated. Okay? So the action potential will be initiated in the initial segment and in the axon hillock. Now there are some diseases where the myelin is a problem. Okay? And sometimes you'll hear me say, what can go wrong? Right? So much of our course is learning about normal anatomy. What's the normal? And you really won't get to appreciate disease or pathophysiology until you go on to the next courses. But we'll cover a little bit of that. So what can go wrong? Well, when myelin is defective, we've got a couple diseases that we probably unfortunately have heard of. And one of them is multiple sclerosis. Did it again. So multiple sclerosis is a degenerative disease, and no longer are you making the Schwann cells in the, back up. You're no longer making myelin in the central nervous system. The problem with MS is that the oligodendrocytes begin to deteriorate in the CNS. This is not a peripheral nervous system problem. MS is a central nervous system issue. And as we'll describe more, the myelin is important for not only protecting the neurons, but also allowing fast, rapid, smooth transmission of the electrical signals. And so as the myelin is broken down, individuals with MS start having what I call hiccups, right? Skips in their nervous system, and they start having issues with motor movement, or maybe later on with some speech or some coordination issues. And the disease is chronic, and more of the myelin begins to deteriorate, and more symptoms are noticed by the individual. Some individuals, we don't understand why, progress through the disease more quickly than others. Some can have MS for 30, 40 years and still function quite well, and others have more of a rapid decline, and the demyelinating events are more frequent, and we don't understand what's going on there. But it's definitely one of those autoimmune diseases. It's we think somehow triggered by a virus, but we don't know. We don't know what really triggers the autoimmune response. It's typically diagnosed in women between the ages of 20 and 40. It is definitely one of those diseases that affects women more than men. Not that men are immune to it, but many of the autoimmune diseases seem to target women more than men.
I had heard in the hospital that when women actually get pregnant that have MS, it actually slows down or stops the progression of MS until the pregnancy is over with.
I wouldn't be completely surprised by that, and I'm doing conjecture. The question was an understanding or a hearing that women who have MS who become pregnant, their MS actually slows down or becomes not as progressive during that period. My guess is that it goes back to the hygiene hypothesis, but I may be wrong. The hygiene hypothesis says this, that 150 years ago, we were living with the animals. We were living in an agrarian society. We were living with more dirt and microbes in our food and with us. And our immune system was developed to fight off those critters. Now we live in a very sterile environment. And we are, antibacterial soap, and everything's pasteurized, and we're not, rolling around in the dirt as much as we used to. And so our immune system, quote, is bored. And so now our immune system is beginning to attack ourselves. So there's some evidence that this idea of hygiene hypothesis makes sense. What some people are doing to fight off or to slow down their autoimmune diseases is to actually infect themselves with parasites. So people are actively infecting themselves with tapeworms. And while that may be uncomfortable, their immune system now has something to play with. So their own immune system now targets their own internal parasite and leaves the rest of their body alone. So I'm wondering, Carl, just out loud, if maybe during pregnancy, the woman's immune system is already dealing with this thing called a baby in her body, and her immune system is already kind of busy trying to figure out, okay, is this thing self or not self? And maybe that is distracting, right? Her immune system from attacking herself and instead dealing with this thing called a baby. I'm conjecturing, there's probably at least 5% truth in there, okay?
If that was the case, then why can't they inject, because they've got like birth control and that kind of thing, why can't they inject them with something like with the human trophic?
And just let them think they're pregnant?
But it's not really, so then it won't progress.
This isn't an issue of tricking the body to think it's pregnant. It's an issue of actually having something that's foreign in your body, right? And your immune system is trying to fight off that foreign object. So just take a parasite. Just go, weekend to Mexico, drink the water, have a few tacos from the street vendors, right? You'll take care of it. Yeah. Okay. Okay. Yeah.
So if they had MS and had, say, rabies or HIV, a double hitter,
Okay.
would having MS slow down
I don't know. Interesting. I don't know if you had some other.
degeneration
I don't know. That's a really interesting. So you're saying, I don't think so. I don't think so. I like that you're thinking, but I don't think so. But you know what? We don't have any short papers due this semester. Maybe I should change that, and you can go do some research on that for me.
of
A few months back for another class, and it said in it that they were giving patients nicotine patches for homeschool children with Tourette's.
That makes sense.
Alzheimer's patients,
Yeah.
and some MS patients, just because of the chemicals that were released from the nicotine would slow down the process of their disease.
Yeah. Yeah. We've all seen people who are addicted to cigarettes, right? And they look like they have Tourette's in between their smoke breaks. Okay, now let's take a look at another disease that goes wrong with myelin is Tay-Sachs. Now, Tay-Sachs disease is a hereditary disease. It's a disorder that is thought to be more oftentimes in a Eastern European Jewish group. However, it also runs in Irish Americans, so it's not just a Jewish disease. But what's going on here is that there's an abnormal accumulation. Tay-Sachs disease, there's a problem with lipids. And what happens is that this abnormal lipid molecule begins to accumulate in the myelin, and the myelin no longer can act normally, right? So there's a problem with the myelin, and these kids die usually by age 4 of all sorts of issues. Part of it is neurological. Part of it is the fact that their lysosomes are no longer doing things quite well. But I just want to point out that there's a number of diseases where myelin is the culprit. So I always want to have you appreciate that what we're learning here is not just random facts that, oh, who really cares? This seems kind of, out there. that this molecule, this myelin, is important not only for your normal nervous system, but when there's a problem with it, we have a disease name for it, and there are plenty of other ones as well. The other one is sort of like MS that attacks the peripheral nervous system and not the central nervous system. Anyone know that disease? Not
It's not Lou Gehrig's.
no, not Parkinson's, not Lou Gehrig's.
What about Addison?
Guillain-Barré. You may have heard of it. I'm not going to have you know it, but Guillain-Barré is a similar disease to MS, but rather than affecting the myelin of the central nervous system, it instead selectively goes after Schwann cells in the peripheral nervous system. Similar, yeah, just a lack of coordination. Okay, so what can speed up the movement of the action potentials? We've talked about, so far we've been talking about anterograde and retrograde. That has not been the signal of the electrical signal. Let me be really clear. The anterograde and retrograde movement up and down the axon was referring to what? Organelles, proteins, vesicles, repair molecules. It was not referring to the speed of the electrical signal, right? The electrical signal can only move in one direction from soma down to the end of the axon. But what can speed up that signal?
Does the myelin speed up both the electrical and stuff in the center?
So Jared's question is, does the presence of myelin speed up the antero or retrograde movement of the molecules inside? No. No. The movement inside the axon is regulated by the microtubules and by the dynein and kinesin, proteins moving up and down the highway. That's separate from the myelin. Okay, so now we're moving over to myelin and talking about the speed of movement of the electrical signal up and down the axon. Or not up and down, just down, sorry. Just down the axon, okay? Now, what can speed it up are two things. One, how big around the diameter of the fiber is. Remember, fiber, a.k.a. axon, right? So how big around the axon is. In lab, you saw a cross-section of a nerve, and you saw in that some axons surrounded by some myelin, and if you think back to that picture, not all the axons were of equal diameter, right? There were bigger ones and smaller ones. So you already have seen a mental picture of this, of different size axons traveling within a nerve. And then the absence or presence of myelin. If myelin is there, it speeds up the signal much, much faster. If it's not there, it'll take a lot longer for it to occur. Okay? And the reason is larger fibers have more surface area. Right? A larger axon has a bigger surface area, and therefore more electrical signals can travel down along that, and we'll understand that here in just a few minutes. What's the speed? Again, this is speed of electrical signals, not speed of organelles or chemicals. So we've got small, unmyelinated fibers. These are going to be the slowest, right? They're not very big, and they don't have myelin. 1/2 to 2 meters per second, not millimeters per day like we were talking about earlier, but meters per second. Okay, that's the slow, right? And as you go up, when you get up to large myelinated fibers, 120 meters per second. What's 120 meters? What is that? Football field, right? So in 1 second, zip, from one end to the other. Okay, that's how fast electrical signals can travel through axons within your body. Pretty remarkable.
Do we have to know these numbers or just the
General
general trend?
trend. So the question is, do you have to know all these numbers? No, but I would like you to appreciate that even the fastest, go back, what was the fastest movement of anterograde? What was it? 400 millimeters per day, right? Which is a snail compared to even the slowest electrical signal, right? Of 1/2 to 2 meters per second. Okay, that kind of comparison. And then appreciate that the larger it is, right? The larger it is, and the myelination will speed it up.
That's only 4 meters, right? A day? 400 millimeters?
400 millimeters per day.
Yeah, that's only less than half. Oh, is it? Oh, it's fast.
Right, right. So we're talking a snail's pace compared to meters per second, right? Okay. Now, where do we have some of the more slower signals? In your body, right? Not all nerves are going to be carrying the same kind of signals. And in your body, you have slower signals going to your stomach. You've all heard it said it takes a few minutes after your stomach's full to let your brain know to stop eating, dummy, right? Your pupils dilate, relatively slow compared to other things in your body. You certainly can't see your eye open and close your pupil to dilate over and over as fast as you can move your finger, can you? Right? So your pupil's dilating a little slower than other muscle action in your body. And then where you need your fastest signals are going to your muscles. So your skeletal muscles are going to have some of the fastest and therefore would be innervated by what? Larger myelinated axons. Signals for balance and vision also are very fast and never stop, right? I mean, you would never want to have any kind of hiccup in your visual signals or in your vestibular signals so that you can maintain your balance and maintain your vision. Okay, so along the lectures, you're going to see these little expected learning outcomes. These are basically little stop and pause when you're studying and ask yourself, am I getting out of this section what I need to get out of it? So refer to these little these ELOs as little review checklists as you're going back and studying. Okay, who's good at Spanish? Who's a Spanish speaker here? Anybody? Okay, so speak very loud this guy's name. Who's my Spanish person? Anybody? Now they're not going to volunteer. Go ahead, Ty. Okay, we got it, right? I just don't want to, ruin his name. So, Santiago, okay? And I'm not going to expect you to know his name, but I want to respect his name properly and not misquote it. But he was a guy who put forth the neuron doctrine. And the neuron doctrine basically says this, that the nervous pathway, our nervous system, is not put together by a continuous wire, but by a series of cells or something in a series connected by gaps. What he said is that it didn't make sense to him that everything in our nervous system was directly hardwired by one wire from the brain to my finger or the brain to my toe. But instead there must be a series of cells or a series of structures in a row with little gaps between them. And what that does is it allows for more control, doesn't it? If we have more spaces along the way, we have more places to control the signal. Okay? So that's his claim to fame, and I just can't pronounce his name very well. So the neuron doctrine brings up these two things. How does a neuron generate an electrical signal? How does that cell send that action potential down? And then once it transmits the signal, what happens next? I mean, what's going on? When the electrical signal goes down the axon and gets to the synaptic vesicle and tells it to release the neurotransmitter, okay, now what? How does this work? And that's where we're going to spend the rest of our time trying to figure out at the molecular level, what is it that's going on when nerves send their signals or when axons send their signals. Questions so far? Always interrupt, just raise your hand, but just want to make sure we're doing okay. So out of this field of research is the area of electrophysiology. That is, we can figure out what are the electrical differences in neurons. Now, I hope that when I said the word electrical differences, already you're thinking, oh, here it comes. Here comes the conversation from lecture 1. Because what did I tell you? That the inside and the outside of the cell have different ions, right? Sodium more on the outside, potassium more on the inside. So there are differences in chemicals, but I also told you there were electrical differences from the inside and the outside of a cell. And I told you that the inside of a cell is more negative than the outside. Okay. So we can take a little electrical probe and stick it inside of a cell and literally measure the difference in the charge between the inside and the outside. And that difference is called the electrical potential. Okay, so there's the word electrical potential, or the polarity, right? The difference from the inside and the outside. And what we're going to see is that this difference is going to allow for electrical currents to be set up, a flow of particles from one point to another. And here's where we get into that sodium-potassium story. So the sodium and the potassium are going to be critical to this current. This electrical current that's traveling through our body is literally the movement of sodium and potassium, opening and closing gates and moving ions along the length of the axon. And this is going to allow us to turn on electrical signals and to quickly turn off electrical signals. So living cells are polarized. What does that mean to you? They're charged. That there's a difference where? Between the inside and the outside. Okay, in charge. Cells that are excitable, especially, are going to be part of this conversation. So what cells have you learned about? What tissues and what cells have you learned about which are in some way excitable? Neurons and muscle, right? Neurons are excitable and they send electrical signals. Muscles are excitable, and when they become excited, they contract. So this conversation is going to be with us now for the next, get ready, we're going to be talking about this for the next month, okay, or at least a couple weeks.
All muscles.
All muscles. All muscles respond to electrical changes, okay? Which is why you can have a little electrical zappers, right, and you feel your muscles contract, right? So we've experienced it in everyday life. Now, we're going to say that the difference between the inside and the outside charge of a cell is its resting membrane potential, the RMP. Okay. And in a unstimulated resting neuron, just a neuron sitting on the shelf, it's going to have a resting membrane potential of negative 70 millivolts. Now, millivolts probably doesn't speak to you, and that's okay. But remember that everything in science, every number always follows with a unit. Okay, rarely do you just write a number. This isn't algebra. So in science, every number will have a unit after it, and the millivolts will be the units we'll be talking about when it comes to the voltage difference from the inside and the outside. Negative 70 tells me what? That the inside of the cell is more negative than the outside, and the difference is 70. Okay, minus 70.
So when they're doing therapy and doing electrotherapy on patients that just had surgery and whatnot, is that what helps stimulate?
They're stimulating muscle. So when a person's had surgery or is going through some physical therapy changes and they do some electrophys or some muscle stim, you're basically using electrical signals to stimulate the muscles involuntarily. Right? You're doing something that the person voluntarily can't do. You're selecting the stimulation.
Build the muscle.
Right, right. Because muscle, just to follow up, if a nerve is cut from the muscle dies. So if you are artificially stimulating the muscle, you will maintain it longer. This is why people who have had spinal cord injuries are going to receive physical therapy on the muscles. They don't have control over them anymore, but they're going to go through oftentimes a lot of physical therapy to keep the muscles doing something so that they won't atrophy quite as quickly. Okay, so negative 70, that's a number that you are going to memorize, that you're going to know. So the resting membrane potential tells me that there's an unequal electrolyte distribution, right? An unequal distribution of negative and positive charges between the intracellular and the extracellular fluid. Does that make sense? Right? There's a difference. And again, there's a 70 millivolt difference between the inside and the outside. Now, how do we get this resting membrane potential? So the resting membrane potential is the result of three things. One, ions can diffuse down their concentration gradient. When something moves down its concentration gradient, what am I telling you? Is it active or passive? Down suggests that it's, I always think of rock going down the hill. right? It's passive. It's going from an area of higher concentration to an area of lower concentration. It's like good old passive diffusion, okay? That's happening, right? Some of these molecules are moving down their concentration gradient. Because where did I tell you? Sodium is hugely concentrated outside the cell. So where would sodium want to go? In. Potassium was hugely concentrated inside the cell, so it would naturally want to go out, just down its concentration gradient. Okay? So that's a little bit of that's going on. Number two, the plasma membrane is in fact selectively permeable. Remember that term? Lab 2, day 1. Selectively permeable means that the membrane has some choice, some selective ability to allow molecules in and out. Some can move easier than others. And then finally, there is going to be electrical attraction of cations and anions to each other. Remember, the cations are the positively charged ions, the anions are the negatively charged. And so remember I told you wherever chlorine is, or wherever sodium is, chlorine is found. Sodium is positively charged, chlorine's negatively charged. They like to hang together, right? So they're attracted to each other by their charge. Okay. So let's talk a little bit more about this resting membrane potential. Potassium has the greatest influence, and you'll see why in a moment. But potassium has the greatest influence on the RMP. And the reason is the cell membrane allows more potassium to just move through it than other ions. It doesn't let sodium in and out quite as easily, but it's a little bit what we say leaky. The cell membrane is leaky toward potassium. So potassium just has the ability to move. Now, which way, therefore, does potassium move? It's found mostly inside. So if potassium was going to, quote, leak, it would be leaking out. What are we losing? What kind of charge are we losing? As potassium goes out of the cell, we're losing positive charges, and therefore the inside is staying negative. Okay, so there's part of it. That negative inside charge is coming because potassium is constantly leaking out into the environment. Okay, and so it's leaking out, leaking out, leaking out. And remember that potassium is very, very concentrated inside than it is the outside. So it's constantly moving out. Now, why wouldn't negative things follow that positively charged potassium? They can't get out because they're too large, or the membrane simply doesn't allow them, right? It doesn't have the same permeability. Many of the negative things in our cells are larger proteins, okay? And they can't get out. So phosphates and sulfates and proteins and those things are negatively charged and they can't get out. Another thing that makes the inside of our cell more negative is DNA. You remember DNA? What makes up the backbone of DNA? Anybody remember that there was the phosphate backbone, right? And do you remember histones? Maybe that when DNA wraps around little proteins called histones to make chromosomes, the histones are positively charged and the DNA is already negatively charged. So DNA is very negative. I'm just saying that as another reminder of why is the inside of the cell more negative than the outside? Because the cell has within it DNA and RNA, and both of those molecules are also very negative. So that's driving that negativity inside the cell. Like Josh mentioned, the potassium gets out easier than other things just because of the way the cell is designed, and so sodium just doesn't have as much ability to go in and out. Sodium is normally found where? Outside, but it does not just flow in easily. Okay, a little bit, but not very much at all. So this is the key line. The resting membrane is much less permeable to sodium than it is to potassium. That's just the way it is. Okay. I also introduced to you, when we were talking about active transport the other day, the sodium-potassium pump, or the sodium-potassium ATPase. And I told you that it does what? It pumps out of the cell 3 sodiums for every 2 potassiums that it pulls in. That is called an antiport. I'll write that down here. And why is that called an antiport? Because as one thing's being brought in, something else is being pushed out. Okay, so it's working in opposite directions. But what is the net result of this? Again, the cell inside is more negative because it's losing more positives than it's gaining. So now we're seeing another reason why the inside of the cell maintains a more negative charge. So here is a little picture of this. And we're outside the cell. This is the extracellular fluid outside, the intracellular fluid here in tan. This is your bilayer, your phospholipid bilayer, your cell membrane. The little orange dots out here are sodium, right? Because we're outside the cell, and sodium is by far the most common ion outside the cell. And we see that the inside is mostly littered with potassium, but it also has larger proteins, which are mostly negatively charged as well. Okay. Now, there are channels here. There are sodium channels and potassium channels. Do you think that sodium and potassium could go right through the membrane on their own? Why do you think that sodium and potassium cannot just move through the membrane? Why do they require this protein channel in the membrane? Tell me about the membrane, the bilayer membrane of the cell. Perfect. What are all these little squiggly chains in here? Hydrophobic, right? Which means that they do not like water, but another way of thinking about it, they don't like polar molecules, right? Water's polar. So water can't go through there very easily because water is polar. Same thing as here. Sodium and potassium, even though they're very small, they can't get through the membrane without assistance because they're charged, they're polar, right? And the membrane is non-polar. So those things don't mix. So we need to have a protein in the membrane that can allow for this movement. And what you see is that look at this. See how busy this is? The potassium channel just really busy. So when I said that potassium had a chance to leak out of the cell, it leaks through this channel. This channel is just a little bit leakier. It allows more potassium to leave the cell. And if you look at the sodium, it's not very busy, right? So there just isn't as much movement of sodium back into the cell. Again, what is that doing? It's keeping the inside of the cell more negative. Okay? Hopefully you're no longer just memorizing that fact, right? That you understand now more reasons why the cell is maintaining that negative charge. What I want to do right there is just take about a 5-minute break and let you guys just kind of regenerate a little bit, and we'll come back and finish up the last hour. But let's take about 5-6 minutes. Any questions so far? Anything up to this point that I can clarify for you? Again, most of what we've seen right now has been much a repeat, a review of neurons and some basic ideas. There's been some new ideas thrown in here. The resting membrane potential is certainly a new one. And if you're making sense of that the inside of the cell is more negative, sodium on the outside, potassium on the inside, and that those ions have to stay in their respective places for things to work, then what I'm about to say will make a lot more sense. Okay, so let's move into this whole idea of potentials. Now, I said there was a resting membrane potential. I'm now going to describe a local potential, and then I'm going to mention an action potential. So the local potentials are little disturbances of the neuron membrane. So, it's just, you got this neuron, and somewhere along the way, it's disturbed, right? There's something that happens, and you stimulate the neuron just a little bit. That's a local potential, a slight stimulation of the neuron. And typically that response will start at the dendrite. It can start instead at the soma, okay? But typically we think of the signal, the local potential at the dendrite, and the signal comes in toward the soma. Or at the soma itself, we can have a local potential, and it's going to travel toward the axon and eventually down the synaptic knob. But let me tell you now, the local potential is not going to go all the way down. Did that wrong again. The local potential is not going to go all the way down to the synaptic knob. The synaptic knob is going to be an action potential. You'll see what I'm talking about in a moment. So when a neuron gets stimulated, it could be stimulated by chemicals, it could be stimulated by light or sight or heat or mechanical disturbance. Remember, there's different kinds of receptors in the body, right? Some receptors are turned on by light, photoreceptors. Some receptors are turned on by chemicals, chemoreceptors. Some are turned on by stretch or pressure, baroreceptors or mechanoreceptors. So it just depends on what kind of receptor it is. But regardless, it is going to initiate or is going to stimulate the neuron. Now, this is where we got to just hold on here. When a neuron gets stimulated, here's what actually happens. Sodium gates open. And sodium is going to rush where? We already know it's mostly where? Outside. So when the neuron gets disturbed, gets activated, gets, stimulated, it's going to cause sodium to come rushing into the cell. Now, what's going to happen to the charge inside the cell? Positives are coming in, so the inside is going to become more positive, or, turn around, less negative. Okay? So we're going to see the sodium is going to flow in. It's going to neutralize some of that negative charge. And so what's going to happen is that the voltage now, the difference between the membranes, what did it start out at? Negative 70. And now the difference will be less negative, and it's going to start going up toward 0 or even greater, up toward a positive number. And we call this depolarization. Now, I don't want you to memorize that word. I want you to think, what does that word mean? Depolarize. No longer as polarized, right? D, to undo. So we're undoing the polarization, we're undoing the difference, and we're going from a negative 70 and we're making that difference less. So we're depolarizing. Now this only happens for a very short while. Okay? So these are very, very quick moments. You're going to think this is this long, slow process because that's how long it takes me to describe it. But these things are amazingly fast. So you're going to get this little tiny stimulation, this little tiny touch, right? This little bit of sodium moving in. And that is a local potential. Okay? So that's the local potential. It does not have a long range effect. It's like a little touch, a little tickle, a little stimulation. Now, a local potential, I haven't even talked about action potentials yet, but this slide will make more sense in a moment. Local potentials are graded. What does that mean? That they can be light to heavy, right? They can have a little bit of stimulation or a lot of stimulation. Or the way I've got it worded here, they can vary in magnitude, right? So I can tickle you a little bit or tickle you a lot, and, a little lighter touch, a little heavier touch, whatever. And the more you stimulate, the stronger the stimulus, the more sodium gates that will open. Okay? So a little touch, a little bit of sodium, a harder touch, more light, more chemicals, more stress would cause more sodium gates to open. Also, local potentials, that's what we're talking about. Local potentials are decremental. That means that over time they can decrease, right? They can decrease in their intensity. They get weaker and weaker the further away the signal tries to spread. Number 3, they're reversible. You can have a local potential that can then be basically wiped out or erased, if you will. And then finally, these local potentials can either be excitatory or they can be inhibitory. And this will make more sense in a moment, but some neurotransmitters are actually going to not cause a depolarization, but a hyperpolarization. Let me just describe this word to you, and then I'll get to your question, Ty. We just said depolarize means what? To make it less different. What would hyperpolarize mean to you? To make it even a greater difference, right? So what we're going to see is that local potentials are graded. They can have stronger or weaker signals. They can be they are decremental. They wear out, they slow down, they decrease. Number 3, they're reversible. And number 4, they can either be on or off switches. They can be excitatory or they can be inhibitory. And in a moment, we're going to see how these 4 features are different from action potentials. Ty?
So isn't it so that you need a certain amount of like stimulation for it to just fire?
That's where we're getting to action potentials. Hold on to that thought, okay? So local potentials are just the initial disturbance, the initial stimulation. We're going to see what the cell does with these local potentials in a moment.
If you constantly stimulate, will it just eventually burn out?
Ideally, no.
Like, for example, when you have a headache, if you have a constant headache, eventually your body regulates or doesn't regulate it, but you still have a headache, but you just don't notice it anymore because you're so used to it.
You're talking about the difference between phasic and tonic receptors. So sometimes the signal will, quote, phase out, or your awareness of it will phase out, whereas other times the signal is tonic and it just never lets go, like a toothache, right? Something just you can't get used to. So that's a little bit different. Hang on, though. Let's get through this local potential and action potential thing. Now, let me show you a picture of a local potential. Try to get an idea of this. What we have here is a neuron. It's spread out from left to right across the top of the screen. So here are the dendrites, many dendrites. Here's the soma. Right here, the trigger zone. Remember I said the trigger zone was found within what? The axon hillock. And then I see all my Schwann cells going down the length of the axon. And then down here, those are not dendrites, but those are instead axon terminals or telodendria. Notice, too, there's no Schwann cell or myelin wrapped around those. So here's what happens. In a little bit of a local potential, you tickle. I'm just going to say tickle. I'm going to keep saying that word tickle. And I want you to understand what I'm saying is you just kind of lightly touch. You're stimulating. You are turning on a neuron. And a little bit of stimulation happens. Now, in this picture, look what happens. Upon stimulation, what's rushing in? Sodium, right? So that's what I just said to you, is that upon stimulation of the neuron, this sodium channel is going to open up, and all the sodium is going to come flying in, because sodium's in huge concentrations out here, and it just comes flying in. What does that do inside the cell? Makes it less negative or more positive. Same thing, less negative or more positive. And where are we? We're in the plasma membrane of a dendrite, right? Typically, we are going to have a local potential start at a dendrite. It also can happen at a soma, right? But it's not happening in an axon, okay? Now, let's compare this with this idea of an action potential. Yeah, can it happen down at the telodendria? Yes. But let's pretend right now it can't. I just don't want to confuse the conversation, okay? Yes, you can stimulate, and we'll get there in a few minutes, but not quite yet. You can stimulate you can stimulate a neuron anywhere where there isn't myelin. Another way of saying it.
Most likely attached to your muscle down here.
Yeah. So let me rephrase it. You can stimulate a neuron anywhere where there isn't a myelin cover, which would be dendrites, somas, and telodendria. Hold on to that, and we'll see what's going on there in a minute.
What about potassium?
Not yet. Not yet. Because it has a different gate, and it's not open. The beginning of this is sodium rushing in. Okay, now, let's compare this local potential deal with an action potential. Action potentials are dramatic. It's like, not a little tickle, but like you got hit with a hammer, right? It's this massive activation, if you will. And when there's an action potential, there's going to be this massive production of very dramatic change in the voltage ion gates. Now, when I say voltage ion gates, right, gates, you got to picture that within the cell membrane there is a protein, a channel, that actually is like a gate. It opens up just like a gate and allows molecules in or out of the cell. So what happens is that in the soma, yeah, there are quite a few sodium gates. There's about 50 to 75 of these sodium gates for every square micrometer. That's just a really small area. Okay? In a very, very small area, there's about 50 or so of these gates. But at the trigger zone, where are we? Down in the axon hillock? There are up to 500 of these sodium gates. So what I'm saying is that you can stimulate a neuron at the dendrite or at the soma, and there will be a few sodium gates that will open up as a result. But it's not that much. But it will stimulate the neuron a little bit. But if you stimulate at the axon hillock, there is a humongous number, that's a scientific word, a huge number of sodium gates that can now drive sodium into the axon hillock. And that will trigger an action potential. Okay? Hang in there. Now, an action potential is only going to be sending a signal down toward the axon, and it will spread all the way down the axon. And that the Schwann cells that we're talking about are going to help protect that signal and assure that the action potential gets all the way down to the end of the axon. I'll show you a picture and it'll start making more sense. Now, an action potential is a very, very rapid, it's a very, very rapid up and down shift in the membrane potential. We're gonna be graphing this in a moment, and you're gonna have this graph absolutely burned on your retina. You're gonna know this graph very, very well by the time we finish this conversation. And what's gonna happen is that there's gonna be this very, very rapid up and down change in the sodium. The sodium's going to come in, and what's the sodium going to do as it comes in? What's that word? As sodium comes in, it depolarizes, which means what again? It's making the inside of the cell less negative or more positive. That's just what happens, right, when sodium comes in. Now, at the axon hillock, if there's enough sodium coming in, that you reach a negative 55 millivolts. Where do we start? Negative 70. If we get up to negative 55, then we say we have reached our threshold. That is, we've reached this point at which all things are going to now turn in a different direction. When you reach that threshold of negative 55, that's another number you're going to remember. When you get to negative 55, we say we've reached threshold and the neuron fires. When that neuron fires, it is creating an action potential. And an action potential is, going back to that list, what was my first one? Graded. It is not graded, right? So go back about 3 slides. What was that slide where I had the local potential? And I said that local potentials were graded, they were decremental, they were reversible, and they were excitatory or inhibitory. Everything on that sheet is wrong for an action potential, because an action potential is not, what was the first one? It's not graded. It's all or none. An action potential is not reversible. An action potential is not either excitatory or inhibitory. It just is. It's excitatory. And finally, what was the other one? Decremental. They do not decrease in intensity as they go down the axon. They are going to make it down to the end in their full glory. Okay? So I'll be comparing that with you in a moment. Question?
So then the local potential is only from negative 70 to negative 55, and once it's negative 55, then it becomes
Good, another way of thinking about it. The local potential is a slight disturbance at a local area that causes sodium to go into the cell, and it travels a short distance. If enough of those local potentials occur, if there's enough stimulation, enough disturbance, such that enough sodium reaches at the trigger point a negative 55. That's when you run, or that's when you hit the action potential. Okay? I'm going to say this about 16 different ways over and over and over. Ty?
So how would that work if you had a bunch of local potentials all over the place? Does that have to be chained together?
So so hold on to that thought. Hold on to it. Josh?
So what's causing the trigger at the trigger zone?
At the trigger zone, there's a huge number, just a higher number of these sodium
I
channels.
know, but what's causing the sodium?
These
expression?
locals.
Huh?
These little local potentials. Okay, so picture you've got a neuron, and you've got 10 dendrites. Okay? And each dendrite just got, quote, tickled a little bit of stimulation locally, right? And each of these little local stimulations are all going to be traveling toward the soma. And they do become collective. So all of these little signals become summative. They become collective. And if the sum of that stimulation reaches the negative 55, boom, then we create an action potential. Now, what if all those signals don't reach negative 55? Nothing happens, right? There is not going to be an action potential because these local potentials degrade, right? They're decremental. So they break down. So you basically have to, stimulate the neuron enough to convince it, if you will, that it has to send the signal down its axon.
Can it be like local potential way down, like with a criminal, for instance, and then no bad difference or something else?
Not yet. Not yet.
So what would cause if the local potential really doesn't do anything because it gets weaker?
Right.
What causes that? And what's the
Whatever is so let's say you're talking about vision, okay? Or let's do something better. Let's do chemoreceptors in your nose, okay? So you've got these receptors in your nose, and they're specific for different chemicals. So a chemical comes along in a very, very small concentration and triggers that neuron, but not very much, right? And so you're not even aware of that odor. But if there were more of those odor molecules and they're coming in and now they're kind of tapping on that neuron and saying at a higher number and causing more little local disturbances, local potentials, then it would reach a point at which it would fire an action potential and now make your body aware of that chemical's presence. That can be true of light, of chemicals, of touch, right? No matter what it is, there's a certain point at which your body doesn't respond, and then there's a certain point at which your body does respond to stimuli. It's the same thing with neurons. Every neuron will pick up some signal, but if there isn't enough of that signal, it won't do anything with it. Okay?
So an action potential is generated by a bunch of localized.
Absolutely. An action potential is generated when there are sufficient local potentials to reach threshold. When that negative 70 goes all the way up to negative 55, boom, now we have an action potential. Good. I think we're getting it. And those who are, you're going to hear this, and this is one of those conversations where you might have to go back and listen to this again. Now, when you're in YouTube, there's a little button, you can listen to this twice as fast. So you can go in and you can hear me talk through it and go to that spot where you know you need to hear it again and then slow it down again. Now, in this action potential, and the graph will come up in after we see the graph, then you're going to go back and look at these slides again. It's going to fill in the blanks. But when that action potential is occurring, what's the first thing? Sodium's coming in, so we're depolarizing. So I'm showing an upward movement with my pen because what's happening to the charge? It's down here at negative 70, and now it's going up more toward positive, isn't it? And it's going to go up, and it's actually going to go even past 0. It's going to go up to 0 and even go all the way up. Now, when it gets to 0, those sodium channels close. They become inactivated. Okay, so the sodium channels just closed. Okay, and they close, but then there's always a little bit more of a change, and the action potential will go all the way up to a positive 35. That's another number you're going to know. Now, what's the charge now? It's positive 35. What does that tell us? That the inside of the cell is more positive. Now, and the outside is more, therefore more negative. Right? We started off with the inside of the cell being more negative, and now in that little area it has become more positive. That, again, is depolarization. Now, here's what happens. Someone asked, what's going on with potassium? Right? You know to ask that. There must be something going on with potassium. I've been talking about it. So we start off with sodium rushing in. Now, once those sodium gates close, now there are going to be some potassium gates that are going to start to open up. They're a little bit slower to open. They're slightly delayed, but they do open up. Now, when the potassium gates open, what's going to happen? Potassium's already on the inside, and when the potassium gates open, now potassium's going to rush out. Now what's going to happen? We're losing positive charges. So now the inside of the cell will become, again, more negative. And what we're going to say is that the cell is becoming repolarized. Right? It's repolarizing. We're going back to where we used to be, where we had a negative 70. Right? We're going to create that polarization again.
Is that what happens at positive 35? That's when the potassium gates open?
Yeah.
So you'll never go above positive 35?
Yeah, the beautiful thing about action potentials is they never change. Right? We're going to say that they're all or none, and we're also going to draw one, and they're all the same. So you'll memorize a couple of numbers, and they're always going to be the same. So, yep, negative 70, negative 55 for threshold, positive 35. Those are the three numbers you're going to remember, and remember them well. Now let's keep going, then I'll show you the picture. Now the sodium gates, keep doing this backwards, the sodium gates are going to stay open a little bit longer, and as a result, a little bit more potassium will leave the cell, and it will actually overshoot. So as the repolarization is occurring, it will overshoot it a little bit, and this little blip in the curve is going to be called hyperpolarization. It's actually going to become more negative. More negative. Now, what's going on here? The cell is actually, or the sodium and potassium are actually changing places. They're switching places, right? The sodium used to be greater on the outside and now is greater on the inside, and vice versa with the potassium.
Because none of the sodium has left yet, right?
Say it again?
None of the sodium has left yet. It's still all in there. It's just potassium leaving.
Well, hold on. This is all one continual thing. So let me get to my next slide. I'm going to go to this slide, okay? I jumped over two. This is the curve. This is the graph that you're going to know to love and draw and trace and just appreciate. Let me try to put what I've been saying into graphical form. So here we are at negative 70. What was that called again? Resting membrane potential. And this neuron is now going to be stimulated by a number of local potentials. It could be one rather large local potential, or it could be a bunch of them all coming in at the same time and acting cumulatively. Regardless, those signals are going to drive, remember those sodium channels open up in response to those local potentials. And at some point, if there's enough of this stimulation, we'll reach that negative 55 number. That's the threshold. And at the moment we reach threshold, there's this amazingly rapid opening of additional sodium channels. Remember I told you that there were more sodium channels at the axon hillock, at the trigger zone. And that's where this initiates. Action potentials start at the axon hillock. And there's this rapid upward swing. Now this rapid upward swing is what's going on. What's happening? Sodium is being brought into the cell in huge amounts. So the cell is becoming less negative, and notice that this is positive up here, right? So we see the curve is going up because we're moving toward more positive. Now at about 0, when we get to about 0, the sodium channels close, and there's always a little bit of overshooting, and we get up to positive 35. That's the other number you need to remember. Now what happens then? at positive 35, now the potassium channels are going to open. But what happens? Potassium is moving from the inside and going out. So what's happening to the charge now? We're going back to being more negative and we're coming back down. This is called repolarization. But it always kind of overshoots. It goes a little bit too low. And it will go down not just to negative 70, but overshoot it by a couple. And this overshoot down here is called hyperpolarization. Okay? We're now hyperpolarized, right? We're even more polarized than we were at rest at negative 70. We're down to about negative 72, 73, somewhere around there. Now, there'll be a period of time where now that hyperpolarization recovers and we come back to where we started at resting membrane potential. Then the neuron can do it all over again, right? Can be stimulated again and fire another signal.
Because we never got rid of any of that sodium that we just took all on board.
Okay, let me tell you what's going on here. You're falling into I won't call it a trap, but a common misconception. And I need you all to close your eyes and just imagine this for me. When I'm talking about sodium and potassium moving in and out of the cell, I'm not changing the entire inside cell. This is hard to imagine, I know. We've been saying that the inside of the cell is more negative than the outside, right? But, and I know I do the same thing. When I imagine the sodium's rushing in, I'm imagining that the entire inside of the neuron is becoming more positive. It's not the entire inside of the cell. It's just a very localized disturbance. I'll try to show you some more pictures of that in a moment. But the whole cell has not become negative or more positive. It's just a little area near where this disturbance was, where this exchange of sodium and potassium has occurred. That's a hard concept. It is, and you'll get there with me. You'll get there. Okay? Not even all the cytoplasm, just the edge near the membrane. Just the little localized area near the membrane. Not the whole cytoplasm, not the whole cell, not the whole axon, just a little tiny bit along the edge. Okay? It may not make sense to you yet, but it will. So you need to know this picture. You need to know this graph. You need to have this one memorized. Okay? So over on the side we have the millivolts. The numbers you need to know are negative 70. That's resting membrane potential. Negative 55 is called the threshold. And positive 35 is always the maximum. It never goes above it. It never goes below it. Every single action potential looks exactly the same. That's kind of nice, right? And that they're all the same. I don't have to memorize different numbers. They're all the same.
Now that one axon that's stimulated, we can't necessarily feel until it's been repolarized.
Say that again for me.
That particular axon.
This is a particular axon, yep.
I mean, once it's been stimulated.
Yep.
And it goes up to the threshold. It can't necessarily feel anything or be re-stimulated.
You're getting, okay, so the question is, I think what you're asking is, once a neuron is stimulated, how soon can it be re-stimulated, right? And that's exactly where we're heading. Those who've already been in lab, all of you been in lab already. You're doing 2 experiments in fills. Number 10 was about action potentials. Remember I told you I wasn't excited about the lab experiments because it wouldn't make sense till we got through this conversation. Now it will make sense to you. The second experiment under fills, number 12, is about refractory period. And that's what you're describing, and that is that once a neuron is activated, there is a time after which it cannot be reactivated. And that time difference is the refractory period. I'll be discussing that in just a moment. And I think I'll get there before we're done. So both of those fills experiments, 10 and 12, will make a lot more sense to you now, after this conversation. So go into those little fills experiments through Connect, read through the introduction, and remember, do the pre-lab questions. Those pre-lab questions will be the exact questions I'll ask you on the quiz next week, and some questions that will show up on the lab exam in 2 weeks. Okay? That was a good infomercial about the refractory period. It's coming up here in just a second. Now I'm going to go back, and I skipped over a slide, and these are videos. They are embedded in your PowerPoints. they will show on your computer if you have PowerPoint pulled up and in presentation mode. You know how when you pull PowerPoint up, if you still have your picture on the right-hand side and you can see all the little files, the little pictures of the other slides on the left-hand side, that's not presentation mode. But if you click on that little projector at the bottom and you get your image in full size, then you can just simply push play. And these things will play for you. Okay? So if I had the hearing on this, you'd be able to hear this guy and walk through it. I'm just going to put these on here as reminders for you to watch and listen to these videos. You'll also see these videos as part of your Connect questions. Many of these videos will be presented to you in your Connect questions. You'll see the video and you'll answer 2 or 3 questions about it. But I've embedded them. I won't always take time to play with them here in lecture, but it's a reminder that when you're going back through this, to look at them and read through them. They will help you. Okay? Now I'm going to go ahead and back this one up, and even though we can't hear it, I want you to see this in time as this goes through. So it's saying this is the cell is at resting membrane potential, and there are sodium gates, and notice that those gates are closed. See how this is closed up here? I think I can do this. Yeah, that's the problem. If I try to write on these, the video stops. So, but we see right here that the activation gate is closed. See the gates right here is closed. It's not open. So it's closed, and this one's closed. All the gates are closed, aren't they? Okay? So that's at the beginning. That's at resting. And what happens is that during depolarization, we see the sodium is coming in, okay? And the sodium is coming in, and it's depolarizing the whole membrane. In a moment, we're going to see now the gates change, and we're going to see repolarizing begin. See how the sodium gates just closed? They became inactivated, and right now those purple potassium gates are still open, and potassium is still being able to leave the cell. It will overshoot a little bit, right, and go to the hyperpolarization. Sometimes that's called the after potential as well. So watch these little videos. They'll run for you really well. And again, in Connect, you'll see questions related to them.
So those gates opening, everything, that's when it meets like the threshold of 35.
When the threshold is met, when the local potentials get up to negative 55, that's when more of those gates open, right? Those sodium gates open. More of them. Okay. See what I need to do to get past this now. There we go. So this picture you need to know really, really well. Okay? Now, let me just give you, I told you a moment ago that action potentials were different from local potentials. Here is that list of facts about that. So action potentials are different from local potentials because action potentials follow an all or none phenomenon. They either happen or they don't. When they happen, they're exactly the same. They will always have threshold at negative 55. They will always go up as high as positive 35. They'll always come back down and overshoot and restore themselves back to negative 70. Always the same, never different.
Is that when things are working perfectly?
This is when even when the body's not working perfectly, action potentials are still working perfectly. Now, there might be myelin or some other issue causing other issues, but an action potential, as far as I know, there's no variation on this theme. Okay? Now, they are also, action potentials are non-decremental. That means that no matter how long the axon is, that the signal will travel all the way to the end of the axon. It will not degrade along the way. Remember I told you local potentials, they degrade. As they come in, the signal weakens. These guys continue that signal all the way to the end. And the myelin is helping to do that. Remember the soma and the dendrites don't have myelin, do they? And that's part of the reason why the signal does degrade. That myelin is electrical tape. It's helping to prevent any kind of degradation of the signal. And once you start an action potential, you can't reverse it. It's going to go all the way to the end. So it's an all or none, non-reversible, always the same, every single time. So looking at this then, which one is which? Well, I don't want to say which one is which. So action potential on the left, that's what we just looked at. Now, what I don't have on here is time. I want you to see what time looks like. Look at this. This spike, right, up to positive 35 and back down, this represents an action potential, and look what's happening. It's happening in less than 10 milliseconds. In fact, most of it's happening in about 2 milliseconds. That's awfully fast, isn't it? Right? 2/1000ths of a second. So this whole thing, this sodium and potassium changing places and thresholds and all this stuff is happening in milliseconds. It's really a phenomenal process. So let's take a look at what's going on with the gates and the channels in the membrane as how it relates to the picture, right, to the graph that we're going to be looking at here. So what do we have here? At the very beginning, in the upper left-hand corner, this is number 1. This is at rest. Okay, the graph shows us we got a flat line down here. We're at rest. Keep doing it, sorry. We're at rest. The line is at negative 70. Okay, resting membrane potential. If we notice the gates are closed, I don't see anything really happening here in the sodium potassium gates. I have a lot of sodium on the outside. I have a lot of potassium on the inside. That's just the way it is at rest. Then number 2, what happens? For something stimulates the neuron. Upon stimulation of the neuron, what happens? Sodium comes in, that establishes a local potential, and we begin to depolarize. Now, if that depolarizing signal is strong enough, and it gets to the trigger zone, and it reaches what number? Negative 55, then that local potential will help form a what? Action potential. And in this curve, we see that it's starting to move up, right? We hit the threshold, we're going up. Then in number 3, we've completely, our sodium channels have opened up, and now look what's happened. They have closed, right? That's the sodium channel. It's not allowing anything in. And now what's happening is the potassium gate opened. And potassium is going to go which way? Out. And what is that going to do to the curve? Bring it back down, right? And that's the repolarizing portion. And then finally, we overshoot it a little bit. We always overshoot just a little bit, come down a little too far. And that was called what again? Hyperpolarization. And now we're back at rest. Right? We're waiting again for another stimulation to occur. And then the sodium potassiums will do their same little dance all over again. Okay. This is one of those areas of physiology. I don't care where I teach and any professor I talk to anywhere on the globe, this is an area that trips students up. And you just have to make these words make sense to you. Depolarizing, repolarizing, hyperpolarizing, sodium, potassium. It's going to take a little while to stare at it, but I think if you just keep coming back to this graph, if you keep coming back to, this graph and drawing of the action potential and ask yourself what's happening at each of these 7 steps, right? Your textbook breaks it down into these 7 steps. So I think you'll be okay with this, but it's not something that most students can absorb all in one sitting and walk away saying they think they get it. It takes a little bit of, knocking a few extra times to get this.
So after your
Polarizing, yep.
and then you reach your RMP.
You go back to RMP.
Yep. And then at that point, does the potassium sodium pump kick in to reverse the...
Okay, so yeah, the question is, so that's exactly, backing up, you're bringing this conversation full circle, which is good. The question is, where does this potassium sodium pump come in, right, that I talked about, the sodium potassium pump, I said it was an active transporter. That is not what's going on here, okay, I want to be very clear. That sodium potassium pump that did the 3 sodiums for every 2 potassiums, That's somewhere off the picture right now. That's off the grid. What we're looking at is a sodium channel, period, and a potassium channel, period, that were part of the action potential story. But what kept the cell at that negative 70 was that sodium-potassium pump. So yeah, in the background, constantly, that sodium-potassium pump is keeping the sodium and the potassiums where they need to be, maintaining that negative 70. Okay? But I can see where the confusion is, right? So when I'm showing these pictures right now, when I'm showing this sodium and this potassium, those are not somehow the sodium-potassium pump. Right? They're off to the right or the left somewhere else. Okay?
So that's happening somewhere else.
That's happening somewhere else on the cell, but not right there where we're talking about it. Good. I saw another hand.
That's just where homeostasis, that's what the.
Yeah, that sodium-potassium pump is maintaining the homeostasis, maintaining that negative 70 resting membrane
and that's
potential.
happening on the other parts of the cell.
That's happening constantly, all over the place.
Even when that part is being triggered.
Yeah. Okay? So just think of it happening next door, but not right there at the same place. Good. Now, I mentioned the refractory period. I think we'll get through this little conversation, then I think you'll probably be saturated. Okay. Maybe you're already there. But let's get through this so you can go do that fills this weekend and feel good about it. So, during an action potential, and for a few milliseconds thereafter, you cannot re-stimulate a neuron. I mean, you can try, right? But there's no way you can get the neuron to fire another action potential.
Stimulated it in a different location.
Yes. Okay? So there's a time at which the sodium and potassium channels are already busy. And I told you this is an all or none phenomenon. So once the action potential happens, the sodium gates open, then the potassium gates open, and that's like a reflex. You can't do anything with it. It's going to happen. It's going to happen in completion. We can't pull it back. We can't reverse it. So it's going to happen. And during that time, now it's only a couple milliseconds, but during that very short time, there is no way you can re-stimulate the neuron. Okay, so let me talk about, this is the idea of refractory period. It's a period of what I'm calling here resistance to stimulation, but there's actually a time where it is not only difficult, it is impossible. Okay? There's actually two phases of refractory period. There is the first part called the absolute refractory period. Now that's shown here in the orangey color. Now what does that mean? Absolutely, no matter how much you try to stimulate that neuron, you will absolutely not be able to create another action potential. And look where we are. Where is this time? It is when the sodium channels are open and when the potassium channels are open, isn't it? It's during that very active time of the action potential. But once we get past it, once we get back down to pretty much the hyperpolarizing, back down to resting membrane potential, then there is instead a relative refractory period. It means that it is going to be more difficult, but it's not impossible. So during the relative refractory period, it is relatively more difficult, but it is not impossible or absolutely impossible to reinitiate. Why do you think they call this why do you think it's more difficult down here in this little phase to initiate another action potential?
More sodium.
You need even more stimulation, don't you? Because you're not starting at negative 70. Down there, you're starting at negative 72, negative 73. So once you get back down to threshold, or not to threshold, once you get back down to negative 70, right, the sodium and potassium have already done their little yin-yang changing of opening and closing. It is now possible to fire another action potential, but it will take even more stimulation because the cell is still in its what? It's still in its hyperpolarizing state. So if you have enough sodium come in, if you have an even greater number of local potentials come in, then yes, you can overcome that difference. You can bring the neuron back to threshold, and you can fire another action potential. So in the very beginning, absolutely not. Absolute refractory period. Then there is a short period of time when there is a relative refractory period where you would need more. It's not impossible, but you would need greater stimulation to cause that action potential. And then once you get past the hyperpolarizing, now you're back to what? Yeah, now you're back just down to good old resting membrane potential, and there's absolutely no restriction or problems with reinitiating another action potential. Okay.
That, the absolute refraction period though, can just literally be a millisecond, can it?
It's a couple milliseconds. Yeah, I mean, it's not enough time you'd even be able to register it in your own head, right? But there is a point at which you just can't do it again. So the words on the next couple slides. So the absolute refractory period, when does it really occur? No matter how much stimulus you give the nerve, as long as those sodium gates are open, you cannot form, absolutely cannot have another action potential. However, the relative refractory period is later on when you still got a few potassium gates open and you're in that hyperpolarizing period, then we're talking about a relative refractory period. Okay? Now, Josh, I think you were asking this, and I don't want to make sure I didn't misspeak. The refractory period only refers to that very small area where the disturbance is occurring. Okay? So you were asking if there's a disturbance in one place, can there be another disturbance somewhere else on the neuron? Yes. Okay? So the refractory period, I may have misspoke. The refractory period does only refer to that very localized area where the sodium and potassiums are exchanging their places.
That pinpoint spot.
In that very localized area.
But if you traveled 10 other pinpoint spots over, you could refire it there.
Yes. Yes.
So an axon could be fired multiple times.
An axon can absolutely have multiple signals coming down the track. Absolutely. Over and over and over and over.
Be fired at a different location.
Right.
Different dendrite.
But typically, though, okay, yes, but yet, where are all those action potentials initiating? Back at the trigger zone. So I don't want you to think that you can be triggering along the axon. You could be continuing to send in signals at the dendrites. You could continue to be stimulating that neuron. And it could send another action potential right down behind the other one.
But it's not. Oh, it is.
You could be sending signals right behind each other. Boom, 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 right down the same axon.
But doesn't the trigger zone need time to recuperate?
A couple milliseconds. Okay, yeah. The trigger zone would also need, because the trigger zone is full of sodium channels. It's the same thing. If the sodium channels are open, you can't pull it back. So yeah, at the trigger zone, there would be a little bit of a delay there as well.
So no matter where, if it's triggered at two different places at the exact same time, only one's going to trigger.
Say that again. If there are two stimuli
In two different places.
of the same neuron, so you're saying two different dendrites are being activated.
Yeah.
If they both come
There's
in,
one thing shooting down the axis.
yes. So let me finish up with that idea. The question or the statement was this: if, let's say, you've got a neuron with 10 dendrites on it, and at the exact same moment, two dendrites receive an excitatory signal. Those signals would do what? Migrate toward the soma and then migrate toward the axon hillock.
Whatever one got there first is going to be the one that goes.
Not even that. They would be summation. They would add together. So those two little local potentials, those two little stimuli would become additive. So, the signal would be added together.
Did you ever have mixed signals then? Like when you're having a heart attack and you feel a pain in your arm, is that what you're
Okay,
thinking?
okay. So we're asking beautiful questions that are going to be great for our next time together, okay? I like where you're thinking, okay? So what I've done is I've given you just enough information about refractory period and action potentials to now go and do Phil's 10 and Phil's 12 before I see you next Tuesday or Thursday for lab. Very good, folks.